Welcome to the Crop Protection Network Spring Webinar Series hosted by the Southern IPM Center. And that's with funding from USDA National Institute of Food and Agriculture Crop Protection and Pest Management Regional Coordination Program. We are one of 4 regional IPM centers supported by USDA NIFA, and we've been charged to coordinate IPM all across the region. I'm Kayla Watson, the Communication Director for the Southern IPM Center. Very happy to be with you today and to help get this great information that's going to be coming at you. It's the final presentation today in the spring series, and it's going to be with our speaker, Carl Bradley. He is a professor of plant pathology at University of Kentucky. So, Carl, really excited about the presentation today, and thanks for being here.
Thank you, Kayla. And let me just say thank you to the Crop Protection Network for the invitation and to the Southern IPM Center for hosting this. This is great. And I was able to catch one of the other webinars in the series, and I'm pretty humbled to be able to be one of the speakers on here. I know that the other speakers were all really good. So I was asked today to speak about fungicide resistance. And so that's really what my topic is going to be about today. See here. Here we go. Before I just dig in, I want to be sure that I do acknowledge some of the people that have provided some funding for some of the research. And so especially a lot of the funding has come from the soybean checkoff programs, either through the United Soybean Board, here locally, the Kentucky Soybean Promotion Board. And prior to me coming to the University of Kentucky back in 2015, I was an extension plant pathologist for the University of Illinois for about 8 years. And so some of the work that you're going to see was funded by the Illinois Soybean Association. And I also want to say thank you to several agrochemical companies and seed companies that have really helped my program by providing some funding and products and other things to help us get along. Obviously, you are well aware of the Crop Protection Network since this is a CPN webinar, but just again, want to show you this webpage. There's a lot of really good information on the Crop Protection Network, and I'll actually show you a couple of the resources that are available there in my presentation today. So the reason that I was asked, I think, to speak about fungicide resistance is because of some of my work on frog-eye leaf spot. And I actually started talking about fungicide resistance even before there was really a problem in soybean that we knew of, at least. And so a lot of it was always theoretical, and then it actually did happen. And so this was from the Illinois Agri-News in October of 2010. And what it's talking about there in the headline is resistance in the frog-eye leaf spot pathogen known as Cercospora sagina to strobilurin fungicides. And so during my presentation today, I'll be talking about strobilurin fungicides a little bit, and they have a couple of other names, and so I'm not, in some places I may be calling it something different. So the strobilurin fungicides are also known as the quinone outside inhibitors, or QOI for short. It's also known as the Fungicide Resistance Action Committee, or FRAC, code 11. So when I'm talking, if you see any of those, we're talking about the same thing, strobilurin fungicides. So frog-eye leaf spot, I would consider it to be the most damaging foliar disease of soybean that occurs regularly here in Kentucky, as well as the surrounding region. Now, there's a lot of different diseases that can affect soybean, and generally the most important ones are those that are down underneath or that are attacking the root and live live in the soil. Things like soybean cyst nematodes, sudden death syndrome, etc. But when we're talking about foliar diseases, I think frog-eye leaf spot is probably pretty close to the top for our region. And the main reason is because of the weather conditions that we have here. So during the summer, we're often very warm, very humid weather. We can get some rainfall. So anything that's keeping those leaves wet, and if we have the warm temperatures to go along with it, those are conditions that are really favorable for the fungus that causes frog-eye leaf spot. This diagram here actually came from a resource that's available on the Crop Protection Network, one that's actually on frog-eye leaf spot. And so this is the disease cycle here. As I mentioned, the fungus that causes this disease is known as Cercospora sagina. This fungus will overwinter in that soybean debris. You can see there on the soil. And so in the leaves and the stems, that's where this fungus can live. And when the conditions are right, when it starts to rain sometime during the summer, conidia or spores will form in that soybean debris. And then when it rains, it'll splash those spores up onto the leaves of the soybean plant, and that fungus will penetrate the leaves. It'll cause a lesion or a spot, and inside of that lesion, it'll produce more spores. And so when it rains again, it'll splash those spores to additional leaves. It'll splash those spores to plants, other plants that are nearby, other soybean plants that are nearby. And so it has what we call a repeating stage. So it continues to infect, produce those spots or lesions, and produce more inoculum or spores that will continue to splash around and infect more tissue. So you can kind of see this repeating cycle here that occurs. And when conditions are really favorable, you can have several cycles within a season, and that's when you could potentially have yield loss. Generally, we don't really start to see symptoms until right around flowering, so what we would call the R1 growth stage. And I'm not exactly sure why that is. Part of it might be that's when we start to see that canopy close, where if you're looking at the rows, where you can really no longer see the space between the rows anymore. And so part of that might be because that's it's giving because of that row closure, you're giving it, or it's providing an environment that's more favorable for infection. So not as much airflow. That leaf tissue is going to stay wet for a longer period of time. So that's when we start to see symptoms normally. And I mentioned that diagram there, that figure showing the disease cycle came from this frog-eye leaf spot publication on the Crop Protection Network. So just looking at symptoms, you can kind of see kind of typical lesions here on some soybean leaves. Oftentimes they may be kind of oval in shape, circular. Sometimes they can be a little irregular in shape. Normally the inside of the lesion, the center, is going to be kind of a lighter color. In this case, it's kind of a light tan color. Sometimes it may be kind of a light gray color. Right around the lesion, there's usually like a dark kind of purplish colored border. So that's what frog-eye leaf spot looks like. Eventually, some of those lesions, the tissue inside of those lesions may fall out. So it can kind of give this shot hole appearance, as you're seeing here. The longer those lesions are out there, normally some of that tissue will fall out that's in the middle of those lesions. So you're losing this the photosynthetic area on the leaves. And so if you lose enough of that, that's going to result in yield loss. Under really severe conditions on a very susceptible variety, you can have really severe frog-eye leaf spot, like what we're seeing here in this picture. This actually came from one of my fungicide trials, where we really try to do some things to give this fungus a really favorable environment, and so we can have some high disease pressure. This is about 50 to 60% of the leaf area that's affected by frog-eye leaf spot. And once it gets to about that level, you can see how some of these leaves are starting to kind of roll up. Eventually they may even fall off. They curl, roll up. And so that's about 60% severity there. So if you're getting to that type of disease severity, that's a real problem. You're definitely going to have yield loss if you have this type of severity. In our fungicide trials where we've seen this type of severity, we've had up to maybe like a 20 bushel yield reduction. So it can be a really important disease under some certain conditions when you're planting really susceptible varieties, and the environment's really, really favorable for infection and disease development. So I mentioned that repeating stage. That's because spores will develop in those lesions, and normally you'll see those spores on the underside of the leaf. So you normally have to flip those leaves over, and you can see them with your naked eye. If you just look inside of these lesions, you can kind of see just a little bit kind of a fungal growth there. Those are actually just spores is what you're looking at. And so it helps if you have maybe some type of a your phone normally is what I do, is just take a picture of those lesions and just zoom in to see it. But maybe you might have a hand lens or something with you to help you see those sporulating lesions. There's a few other things that can cause similar type spots. So it's always good to be able to get a good diagnosis of what is causing the problem. So if you're seeing these spores on the underside of the lesions like that, very likely it's frog-eye leaf spot that you're looking at. Just a close up there of those spores. So every year since 1996, plant pathologists like myself from lots of different states will get together and estimate the amount of disease loss or yield loss caused by different soybean diseases. So what you're looking at here is the estimated yield losses caused by frog-eye leaf spot from 1996 to 2018. So this is not completely updated. The 2019 data, I think, are now on the Crop Protection Network, but this is up through 2018. I just wanted to show you that here in the last few years, since about 2013 or so, we've kind of had a jump in yield reduction caused by this disease. And you can see what happened in 2018. So in 2018, there was just a lot more frog-eye leaf spot showing up in areas where we never really thought it was much of a problem before. So this is a disease that is kind of maybe on the rise a little bit, where we're seeing it cause a little bit more yield loss than what we thought it used to. What I'm showing you here, these are just those yield loss estimates in percent. So percent yield loss to frog-eye leaf spot. This is actually 2009, and you can kind of see the estimates state by state. I'm going to circle the ones that are at least 1% yield loss. Okay, so in 2009, it was in Tennessee, 5% yield loss, kind of where we would think it would be. This region around Kentucky, in Tennessee, you know, that's going to be an area that frog-eye leaf spot is generally going to be around, causing some yield loss. So that's 2009. 2012, again, kind of in the southeast where you might expect it. 2015, now you're kind of seeing it in other places. You're seeing it out east a little bit. And then 2018, where we saw that there was such a big problem that year. Not only is it in the southern mid-south where I am now, but places like Illinois, South Dakota, Iowa, areas that typically have not generally encountered big problems with frog-eye leaf spot, starting to see that happen now. So it looks like this disease is one that's starting to be more of a threat in states outside of where I'm located. So states that are north of Kentucky are now starting to experience more problems with frog-eye leaf spot. So as far as management of this disease, there's different things that farmers can do. We kind of have our basic things here that a lot of them are already doing to some degree. Crop rotation is important because this fungus overwinters in that soybean debris. So if you're growing continuous soybean, especially like maybe in a no-till situation where you're leaving that soybean debris there on the soil surface, right where that inoculum could develop and then just easily splash up onto the new soybeans that are coming up. That could be a serious situation where you could have a lot of disease. So crop rotation can help break those disease cycles to some degree. There are resistant soybean varieties in some areas. Now, as you start to go further north, it's likely that seed companies have not probably been intentionally breeding for resistant varieties in places like South Dakota. So there may not be really any resistant varieties in a place like that. But here where we've been dealing with frog-eye leaf spot for a number of years, there is a range of susceptibility across the varieties. And so we can find some varieties that are going to have a high level of resistance that are available in like maturity group 3s and 4s. And then foliar fungicides, which is what I'm going to talk a lot about today, has been a part of frog-eye leaf spot management program for a number of years now. So when we think about the different fungicide classes that are registered on soybean, here's 4 of those classes. We don't have a whole lot when it comes to foliar fungicides. We have a few more than these, but I'll show you these because these are ones that tend to have some efficacy against frog-eye leaf spot. So we've got the MBCs, or FRAC code 1, listed here, the methylbenzimidazole carbamates. And the other thing I have over here in the far right column is the risk of resistance developing. So that FRAC, that Fungicide Resistance Action Committee, has provided these ratings for all of the different chemistry classes as far as how, what type of risk, intrinsic risks exist for fungi developing resistance to that chemistry class. So you can see this FRAC code 1 or the MBCs has a high risk of fungi developing resistance. Then we've got our FRAC code 3. We usually call these triazoles, but the official name is demethylation inhibitors, or DMIs. And you can see those have a medium risk of resistance developing. And we kind of have this class that we're starting to see as we have newer fungicides registered here over the last 2 or 3 years. A lot of those are including an SDHI fungicide, or FRAC code 7. And you can see that those have a medium to high risk of resistance developing. And then we have our FRAC code 11. So those are the strobilurins, or the QOI fungicides. And you can see that those have a high risk of resistance developing. So that FRAC code 11, that's the one I'm going to be talking a little bit about because of the resistance is occurring in the frog-eye leaf spot pathogen. Within this, this fungicide class, there's actually 3 different mutations that are known that it can occur in fungi that will give those fungi resistance to that chemistry class. This top one here, known as the G143A, that's the most common. That's the one that we observe most often when there is resistance that occurs to this fungicide class. That's the one that we see the most. That's also the most important because that provides almost complete resistance to the fungicide. But you can see all 3 of these mutations that can occur. They're just simple shifts in the fungus. It's just simple amino acid shift. So it's just a one-step process for the fungus to develop resistance. And that happens because fungi, just like humans, animals, everything basically that's living, are going to mutate to some degree naturally. Those mutations just occur naturally. And so in fungi, the approximate rate of mutations occur in about 1 in 100 million. And so those mutations occur. Some of those might be mutations that confer resistance to a fungicide, and they just exist naturally. Once we start to see problems is when we apply that fungicide. And so we're starting to apply that fungicide, and we select out those individuals that have resistance. And so those now become the normal, what we see the most of. And so that's kind of how fungicide resistance ends up developing. So normally the first step, if you want to monitor for resistance, which is some of the work that I've done with the frog-eye leaf spot pathogen over the last few years, the first step is to develop that baseline sensitivity. So we want to know how sensitive was this fungus population before we ever started using the fungicide. So that's usually the first step. And so I was lucky enough to have access to some isolates of the frog-eye leaf spot pathogen that had never been exposed to QOI or strobilurin fungicides. These were ones that were collected, anywhere from the late 70s to the mid 1990s. So time before we were using strobilurin fungicides. So I was able to get my hands on those, and we were able to do some lab tests. So what we do is we, in Petri dishes, we have some media in which the fungus will grow on. And in that media, we add different concentrations of fungicides. And so we can put the fungus on that media, and then we can measure how much it has been inhibited. And so with this particular fungus, we used the spores, and then we measured if they germinated or not. And so this is just a picture here of, on your left, a germinated spore of Cercospora sogeina and one that was not germinated. So if you look at enough spores, you can get an estimate of how many were affected by the fungicide that it was taking up through that growth media. And when we do that, we can calculate what we call an EC50. So EC50 stands for the effective concentration at which 50% of the spore germination is inhibited. So the higher the EC50, that meant the more fungicide it took to get a 50% inhibition. So we did this with some different QOI fungicides back in the late 2000s. And this is just showing, we did it with azoxystrobin, pyraclostrobin, and trifloxystrobin. So this was the level of fungicide it took to get a 50% inhibition on isolates that had never, ever been exposed to this chemistry class before, okay? So that was our first step. So then after that, after we knew what that baseline sensitivity was, the next step was to go out and collect isolates of this fungus from fields that had been applied with a QOI or strobilurin fungicide. And I got some funding from the Illinois Soybean Association to do this work. So we started in Illinois, where I was located at that time. So from 2007, 2009, we focused on Illinois. We got our methods down and felt pretty good about what we were doing. So we put out a request to other states in 2010. We asked like Indiana, Kentucky, Tennessee, other places, say, if you find any frog-eye leaf spots, send us some leaves and we'll do some testing. So in 2010, we started doing that. And we basically got the leaves into our lab, and we would get those lesions to sporulate. And from there, you could see the spores, and we would pull off single spores and grow them in a Petri dish. And we would do the test and look at, calculate the EC50 and see what happened. Just doing the spore germination assays. And so this is just an example of trifloxystrobin. Here's our baseline level. These are our baseline isolates. 2007 isolates we collected in 2007 looked about the same, pretty much the same EC50 level, which is fine. That means nothing's really happening. Then we got 2008 isolates, and you can see 2008, 2009, you can see how it's slowly starting to creep up. So again, the higher the bar, higher the EC50 level, that meant it took more fungicide to get that 50% inhibition level. Okay, so we were starting to see that the fungal population of Cercospora sogina was starting to become a little bit less sensitive over time. And then we came to 2010, and you can see what happened in 2010. Okay, looking at our baseline here, it was 0.0011. In 2010, the average of all the isolates that we collected that year was 0.6835. So we saw over a 600-fold shift in sensitivity in the average EC50 from isolates that we collected in 2010. So that's not good. That means that basically we had resistance occurring in some of those 2010 isolates. So when we started looking at it by isolate, we found that the ones that were causing that high EC50 came from that circled area there. So there were two fields in southeastern Illinois from a couple of different counties there. One, actually right where I'm sitting right now, Caldwell County, Kentucky, and then a couple from West Tennessee. My colleagues at that time had sent me samples from Tennessee and Kentucky, and we found resistance in those five fields in those 3 states. Now, the interesting thing, when we did this work, we were able to look at how many of the isolates were still sensitive, the fungicide still worked, and how many were resistant from each of those locations. So this is just sort of a snapshot of several different fields that we collected from, or people sent us isolates from, or samples from. And, if you look over the right, anywhere from these 2 Indiana fields to Warren County, Illinois, and Northwestern Illinois, all of those, all the isolates we got were still sensitive, okay? So if you focus on these 5 on the left, those are those 5 fields where we found the resistant isolates. So the resistant one, the percent resistant isolates are in orange, and the percent sensitive are in blue. If you just focus on these 4 here, the Gibson, Tennessee, Caldwell, Kentucky, Pope County, Illinois, Gallatin County, Illinois, you can see that the majority of what we were finding were still sensitive. So the blue bars were a lot bigger than the orange bars there. But we had this one field in Lauderdale County, Tennessee, where 100% of those isolates that we got from those leaves were resistant. So we did some checking just to kind of see what the deal was with that particular field. And that was a field that was in the sort of planted in the river bottoms in West Tennessee. And just because of the water always being up high, that farmer could never really plant corn because the water went down late and it would have been too late to plant corn. So he had basically had to plant continuous soybean. So that sort of was your increase in inoculum there. Always a lot of inoculum. Of course, it's in the river bottom, so you know it's going to be very humid in Tennessee. So that farmer had a problem with frog eye leaf spot. So to solve that problem, started spraying fungicides. And those fungicides generally in the mid to late 2000s, at least, a lot of those were strobilurin fungicides, oftentimes just strobilurin fungicides, not mixed with anything else. And so what happened was over time, as that farmer sprayed strobilurin fungicides, all those isolates he was selecting out were resistant. Okay, so that's what happened over time. We were able to develop some quicker methods, so we didn't have to plate out lots of different plates and calculate EC50s. We were able to calculate that if we used 0.1 part per million of azoxystrobin, that could show us if it was resistant or not. So just as an example here, on the left, these plates here don't have any fungicide in it. So that's the fungus growing just fine. The one in the upper right-hand corner, that one is a resistant isolate with azoxystrobin at that 0.1 part per million concentration. And you can see it's growing just fine, and our sensitive isolate's not growing. So we can use that dose, just one dose now, to determine if it was sensitive or resistant. We also were able to do some molecular work and find out that what the mutation that was responsible for this resistance was that G143A mutation that I talked about earlier. It's just that single nucleotide shift. And as I said, it's the most common mutation that's found in fungi with resistance to this chemistry class. And this mutation makes that fungus almost completely resistant. So that's not great that the fungus has this mutation, because it basically means that strobilurin fungicides just don't work at all anymore when it has that mutation. We were able to develop some molecular methods to be able to identify this mutation a lot quicker. So once we had all these tools in our toolbox now as a research lab, we could start to examine a lot more samples, a lot more isolates. And so, from 2010 on, we started doing that. And then some other labs started doing that as well. So not just the Bradley lab, there's other labs around the US that are also doing some of this work and looking for these QOI resistant strains of this fungus. So I'll just show you a map here. This is 2010. You can see those 5 fields in Illinois, Kentucky, and Tennessee. And this is up to 2013. And so my lab, as well as some other labs, started to identify it in a lot of other places. This is up through 2016, 2019, and now you can see it's definitely up in the Midwest. You can see Iowa. Dr. Darren Mueller there at Iowa State sent me a bunch of leaf samples and isolates from that year, and we were able to confirm a lot of that. You can see Nebraska, South Dakota is now included, Minnesota now included there as well, Michigan, Ohio, lots of different places. Most recently in 2020, we have now added Wisconsin and North Dakota to the map. And North Dakota is actually very surprising. Having spent some time there back in the early 2000s, I was an extension plant pathologist in North Dakota. Never would have thought frog eye leaf spot would have been a problem there, just based on your normal conditions that occur. It's usually just not quite humid enough or warm enough for frog eye leaf spot to take off. So apparently things are changing. And so it did show up in 2020, the disease, but also these strobilurin resistant strains were there. So we've now confirmed this in 21 different states, and you can see the number of counties and parishes there. Now you see a lot of, like if you look at Illinois, if you look at part of Kentucky, Missouri, et cetera, there's still a lot of blank space there. That doesn't mean that these QOI resistant isolates are not there. That just means that we never got a sample from those counties to test it. But pretty much what we're finding now is if you have frog eye leaf spot, I will just about guarantee you that some of those isolates that are causing that disease are going to be resistant to strobilurin fungicides, no matter where you are in the US. And if you remember, I showed you some of that data from 2010 and some of those fields, if you forget about that Lauderdale County, Tennessee one, some of those other fields, the resistant isolates were in the minority. Mostly we were finding sensitive isolates. Pretty much now, what we're finding is there's been a complete shift. When we find frog eye leaf spot, it's generally going to be the strobilurin resistant strains that are present. So these are just some information here from Nebraska in 2019. Dr. Tamara Jackson-Zim sent me some samples and we were able to confirm the strobilurin resistance. And this just shows you, this table here shows you the different counties and then the number of isolates that we got that were resistant, the number that were sensitive. But if you focus here on this percent resistant, a lot of these were 100% of the isolates were resistant. It was only just a couple of fields, a couple of counties where we saw that it wasn't 100%. And then it was like 80 to 90%. So what we're finding now is pretty much if you have frog eye leaf spot, the majority of those isolates are going to be resistant, even if you're finding it for the first time in your state or wherever in your county, doesn't matter. They're out there and that's what we're dealing with now. So I'm going to show you some data now from some fungicide trials that I've conducted here at the University of Kentucky. We've been doing fungicide trials for quite a while now, but I thought I'd show you a little bit more recent data that would include some newer products. So the results of the trials I'm going to show you came from 7 site years, and you can see the locations that we had here. I'm located here at Caldwell County, Kentucky, at the UK Research and Education Center. Did some trials in a county just north of me on a farmer's field, and then UK has another research station in Eastern Kentucky that we do some trials. So there's not a lot of soybeans out here, but the conditions are very favorable for frog eye leaf spot. You have a lot of hills and valleys and a lot of very humid temperature or humid environment, warm temperature, almost perfect for frog eye leaf spot. So we do a lot of trials there. So we do some things to be sure that we get good frog eye leaf spot. So when I talk to growers, I tell them, you're not a plant pathologist, right? You're a farmer. You don't want to do these things. So do the opposite of a lot of this. So when it comes to picking out the varieties that we want to plant in these trials, I'm like the opposite of what most people do. I'll look through seed company catalogs and I will look for the varieties that are rated to be the most susceptible because I want to have a good disease pressure out here to be able to evaluate these fungicides. The other thing I do is I grow soybean on soybean. I don't rotate in a lot of these research trials because I want to build up that inoculum. We plant no-till, so we don't till the soil. That leaves that soybean debris on that soil surface. And basically those spores are just going to be right there. So when it rains, it's going to splash those spores up onto those soybean leaves. And for the results I'm going to show you, these were all fungicides applied at the R3 developmental stage, which is when those pods are just starting to develop on the soybean plant. We've done some work looking at some different timings, and overall what we find on average is that R3 growth stage is what tends to give us the best control during the most crucial development time of soybean plants. So that's usually what we're targeting here. So here's just the average across all those 7 site years. What you're looking at here is frog eye leaf spot severity. So the higher the bar, the more disease was there. We do this just by going back out about 3 to 4 weeks after we make those fungicide applications and evaluating, especially the leaves on the upper third of the canopy. And that's just an estimate of the percent of the leaf area that's affected. So here's our untreated check. You can see it's about 37% severity. This is headline fungicide. That's a strobilurin fungicide. So you can see it's not working very well because of the fungicide resistance. And I'm going to bring this in here just to kind of help us out a little bit. So that's our QOI or strobilurin fungicide headline. Then I've got some fungicides here that some of these are mixtures. Some of these are just straight DMI or triazoles. And you can see that they're working. We did bring that severity level down. We also find that the FRAC group 1 or the MBC fungicides are working. Some of these are like Topsin is just a straight MBC fungicide, whereas things like Froghorn and Acropolis would contain an MBC and a DMI fungicide. And then some of our newer fungicides, as I mentioned before, some of those include an SDHI. So some of these are mixtures of triazoles with an SDHI, or maybe a three-way mix with a strobilurin as well. So we are getting good efficacy, okay, with some of these other products. So, you know, we're still in good shape. But the thing you have to remember is that these other active ingredients, these other chemistry classes, there's also risk for resistance developing to those as well. And so for right now, we seem to be okay, but things could change. And just because we have a lot of, you know, pretty high severity in our trials, we're able to get a pretty good yield response. Now, you wouldn't always, you know, in a typical farmer field where they're picking good varieties, they're rotating, you wouldn't necessarily see this type of a yield response with the fungicide. But in these really high disease pressure situations, you know, we can measure up to, you know, in this case, around a 20 bushel yield response from our untreated to the, you know, the highest yield that's here. So in summary, from that series of trials, you know, we found that those strobilurin fungicides are just not effective in controlling frog-eye leaf spot in our region anymore. But the DMIs, the SDHIs, and the NBCs still seem to show efficacy, so that's good. But as I mentioned a minute ago, there's still a medium to high risk of resistance developing to these chemistry classes. So we still have to be careful. Now, what can we do? Well, I think what that means is to not apply those on their own. So we really need to either tank mix or use fungicide products that have at least 2 active ingredients from 2 of these chemistry classes. Okay, so we'd want to use something like a DMI mixed with an NBC or an SDHI mixed with a DMI, whatever. We want to be sure that the 2 active ingredients that we have, both of those have efficacy. Okay, that's going to help slow down the selection for resistance. Now, to help us make some of these decisions on foliar fungicides, on the Crop Protection Network, there are the fungicide efficacy tables, where there's one for soybean foliar diseases, as you see here. And actually, this is what, April 6th, I think. A few weeks ago, the new version of this table was uploaded onto the Crop Protection Network site. I'm showing you the a little bit older one, so I apologize for that. There is a newer one that will look slightly different here, but it's going to give you similar information. So when you look at this table, if you open up that publication and look at this table, you can see across the top here, it gives you all these different diseases, and on the side here, it gives you some different chemistry classes and some different active ingredients and products. If you look at frog eye leaf spot and the strobilurin fungicides, you can see all those are rated P, and that P does not stand for perfect. That stands for poor because of the fungicide resistance that's occurring to that chemistry class. If you look at the triazoles, there's a range of fair to very good. We only have one active ingredient that's in the NBC fungicides, that's thiophanate methyl, and it's shown to be very good. Now, you would not want to apply this product by itself, though, because it only contains thiophanate methyl. And we know that chemistry class has a high risk for fungicide resistance occurring. So we would always want to pair it with a triazole or something that also has good efficacy. Here's the SDHIs. Now, we only have one that has one SDHI all by itself. That's Boscalid or Endura. That's not really, it just doesn't work for frog eye leaf spot. That's a good fungicide to use for like Sclerotinia white mold if you're in a state that's a little cooler where that can be a problem. But that one in particular just doesn't have the efficacy. But there's other SDHIs that do, and you can kind of find those here in some of these mixed mode of action chemistries. The new version of this table will provide the different chemistry classes over here on the left. So that's going to make it even more helpful. But just as an example, this one here, this Lucento, it contains a triazole and an SDHI. And so you can see that has very good efficacy. And there's others here that contain a mixture of chemistry classes that you're going to get very good efficacy. So we have a lot of options, fortunately, and we can take mix if we need to. One other series of trials I'm going to show you some results from here real quick. This was funded by the Kentucky Soybean Promotion Board, trials that we conducted in 2016 and 2017, along with the University of Kentucky Soybean Performance Variety Trials, that are coordinated by Dr. Claire Vennard. So you can see all the different locations that we had here across the state in both 2016 and 2017. And in this case, we were integrating two management practices. We had the fungicide component, but we also included the variety resistance component. So we had three different varieties. We had one that was rated as being pretty susceptible to frog eye leaf spot, one that was rated as being moderately resistant, and one that was rated as being pretty highly resistant to frog eye leaf spot. On top of that, we had three different fungicide treatments. I wish we could have had more, but just because of all those locations and we were kind of limited on space, we had to just limit it to three different treatments. And so we had an untreated check, and we used headline because we wanted to just have a solo strobilurin product in there to see if resistance was occurring. And then for the product that we used that had good efficacy, that was a product known as Quadris Top, which was a strobilurin triazole mixture. So what you're looking at here is similar to our fungicide efficacy trials I showed you a few minutes ago. This is looking at frog eye leaf spot severity. So the higher the bar, the higher level of disease there was. Down here at the bottom, we have our susceptible variety, moderately resistant variety, and resistant variety. The blue bar is our non-treated check. That black bar is our headline, our strobilurin fungicide. And the gray bar was our fungicide that was effective, that Quadris Top. So the only place you can see that asterisk is in that susceptible variety. So if you look across here on moderately resistant, there really wasn't enough disease there for that fungicide to really even provide much of a benefit as far as controlling frog eye leaf spot. And just as an example, these were actually pictures from the Murray location in Western Kentucky, where we had on the left, we had our susceptible variety, and on the right, we had our resistant variety. So, you know, there are differences, big differences across different varieties. Some universities will rate for that. In our area, the University of Tennessee, Dr. Heather Kelly does some ratings, and that goes into their annual publication. This last year in 2020 at the Hancock County location in Kentucky, we were able to have a high level of frog eye leaf spot for us to go out and provide some ratings. So we've got ratings in there. And of course, your seed companies are generally going to provide some ratings for frog eye leaf spot. So there's lots of places you can find information on how susceptible a particular variety might be. When we looked at yield, this is what we saw. So this is yield. The higher the bar, the more yield there was. If you looked at the moderately resistant and resistant varieties, pretty much even across those, just because there just wasn't a lot of disease present. And so we didn't see very much of a response with those foliar fungicides. However, in that susceptible variety, because there was frog eye leaf spot on that susceptible variety, we did see a significant response, yield increase with the Quadris Top because of its efficacy against frog eye leaf spot. So in general, you know, that solo strobilurin product did not really provide efficacy because of the fungicide resistance that occurs in our state. In general, the largest yield response that we saw was on the frog eye leaf spot susceptible varieties. And of course, that occurred when we used a fungicide that had efficacy against frog eye leaf spot. So, you know, we could have about 4 more other presentations on fungicide resistance that would not just be talking about frog eye leaf spot, talking about other pathogens. So this is occurring in other pathogens that cause disease of soybean. For instance, the Cercospora leaf blight pathogen, Cercospora flagellaris in Louisiana, they have found resistance in that pathogen to both the QOI and the MBC fungicides. Septoria brown spot, that pathogen, Septoria glycines, we're starting to, some work that we're doing, starting to find resistance in a lot of different states, including Kentucky, Tennessee, Iowa, Illinois. So different areas where we're finding resistance to strobilurin fungicides within that Septoria glycines pathogen. Target spot is a disease that's kind of an emerging disease here in the lower Midwest or Mid-South in states south of us. This is caused by the fungus Corynespora cassicula. And in Alabama, they have reported resistance to QOI fungicides. And I know there's some other places doing some work on that. And so it's likely to be in other places besides Alabama as well. And we've been looking at this in Kentucky. So far, we have not identified QOI resistant strains in our state yet, but they're likely out there. We just haven't found those isolates yet. And then finally, also in Louisiana, the aerial blight, Rhizoctonia aerial blight fungus has resistance to the QOI fungicide. So this is a growing problem where we're finding this on a lot of the different foliar and stem pathogens that can occur on soybean. I mentioned the Crop Protection Network. That's a really good place to go for information for a lot of different diseases on different crops. We do have some publications, one in particular here on fungicide resistance. So it's just fungicide resistance in field crops, FAQs, frequently asked questions. That's available. I'm also involved in another activity with this Take Action group. And if you go to iwilltakeaction.com, there's also some good information there about fungicide resistance. So what can we do? Is this a problem that just is always going to happen and there's nothing we can do about it? Well, to some degree, it is Mother Nature, and Mother Nature generally finds a way to kind of overcome what we want to do. But we can help slow down this fungicide resistance. So how can we do that? Well, we can do that by not just relying on fungicides. There are other ways to manage a lot of these diseases. So thinking about integrating those other disease management practices into our operations, things like crop rotation, resistant varieties, those are all can be very important methods in reducing disease levels. So not just relying solely on fungicides to control diseases. And then we should also only be thinking about applying a fungicide when it's necessary. And so what I mean by that is when we have a disease to control, when we are based on our scouting observations, or if we're doing some type of a risk assessment, if we know that, we've got a susceptible variety, we've got the conditions that are favorable, we've got, maybe we didn't rotate, things that put that risk a little bit more towards diseases being present. Those are the things we should be looking at. So if we're just applying because we want to, we just want to increase yield, but we're not really paying it, maybe we planted a really resistant variety, maybe we don't need to spray a fungicide. So limiting those fungicide applications to only when we really need it for disease control. And when we make that decision to apply a fungicide, it's important to not use just a single active ingredient fungicide. We got to use a product or tank mix a couple of other products that's going to contain different active ingredients from different chemistry classes where both of them have efficacy against that target disease that we're wanting to control. So with that, I just want to, I've got the names of some members of my lab up there. A lot of this work just wouldn't be possible without the people that are really doing all the hard work there. And so I appreciate their help. And I'd be happy to try to answer any questions.
All right, thank you, Carl. That was an excellent overview of fungicide resistance and frog-eye leaf spot. For those of you who don't know, I'm Kirsten Wise. I am a plant pathologist also at the University of Kentucky. And I'm just going to sort of moderate a Q&A with Carl. And Carl, we have lots of questions that have come in. So I'm just going to go ahead and jump in and start asking some of those, okay? And if any of you have some questions while we're talking, please submit them through the Q&A. All right, so the first question is about frog-eye leaf spot and distribution. So you showed your nice maps of how it started out, and we're seeing some yield loss in the southern states, and it's really spread up into those northern states. Why do you think frog-eye leaf spot is more widespread and yield limiting than it used to be?
I don't have an exact answer for that, but I can speculate, I guess. I think there's a couple of things. I think that the fungicide resistance maybe could have played a role to some degree. When you're thinking about just microbes that are competing out there in nature, that could give this one a little bit of advantage. If something is sensitive to the fungicide and this isn't, it might help it survive. I think environment is probably a big part of it. I mentioned, I used to be at North Dakota State back in the early 2000s. And at that time, I never saw a summer where I thought frog eye leaf spot would be able to be present. And obviously last year, talking to my colleague, Dr. Sam Markell up there, that they were pretty humid and they had conditions that are really favorable. So I think just overall, our climate's changing, and some of these diseases that used to be kind of limited to a certain area, we're probably going to start seeing them in other places.
Right. Yeah, I think that's right. I think the climate has a big part of it, right? So the next question would be, how severe does frog eye leaf spot have to be before you see yield loss?
That's a great question, Kirsten. And it's one that we don't necessarily have hard data on. I will say from my fungicide trials, and I actually have a graduate student that's working on this, and he's not quite there yet, but I've been able to look at some of his preliminary analyses. It looks like if you are getting to about 10 to 20% severity, it's likely you may have some yield loss there. So that would probably be, your trigger might be, somewhere between 5 and 15%, maybe, if you're out looking, but timing is everything, right? So is this like 15% when you've got, you're almost at R8 where you're completely mature? No, this would be, you'd have to back that up at maybe like R2 just after flowering. So it's a little bit, we don't have exact numbers on that, but that's something we're hoping to get to.
Right. And would that depend on variety too? I mean,
oh, absolutely. Yeah. So it's also important to know like how susceptible your variety is. So, you need to know that. And I think that's a big part of the decision making on spraying a fungicide or not. So maybe you went for yield. Maybe you just like picked the variety that had the most yield potential, and you really didn't care about the other traits. You didn't care about how resistant it might be to frog eye leaf spot. And so, if you planted a really susceptible variety, that's one that you're really going to have to watch. You're really going to have to target and watch. And if you're starting to see disease develop and you're at flowering and it looks like the weather is probably going to continue to be wet or warm, whatever is sort of driving it, that's probably one you might want to spray. Whereas if you planted a variety that was, let's say if it had a 1 to 9 rating and 9 meant the most resistant, if it was like a 7 through 9, it's probably not going to get that much disease. You could probably just leave that one alone.
That sounds like my kind of variety, the one that you can just leave alone. I like those. So this is a good question about some of those disease loss estimates. Do you think there's any confusion between frog eye leaf spot and Phyllosticata when they're doing those estimates?
That's a great question. So I actually, in one version of this presentation, I had some pictures of Phyllosticata, but I took them out because of time. So Phyllosticata is another disease that we're finding that looks almost exactly like frog eye leaf spot. There's just one difference. There's some what we call pycnidia that will develop. There's little dark structures that will develop in the lesions, which can tell it apart from frog eye leaf spot. Normally with Phyllosticata, though, we're finding those a little earlier than we find frog eye leaf spot. So to answer that question, I think it's possible, but on the other hand, the people making those estimates are plant pathologists, and I feel like they're probably doing a good job of knowing what's what. But you never know; some of those could be causing some of that, maybe.
Yeah, and I think just general education for farmers, right, about the difference between those two diseases is really important because they do look a lot alike. I've been confused by them.
Yeah, so that Crop Protection Network publication on frog eye leaf spot has some good pictures of Phyllosticata in there as well, and some other things that look like frog eye leaf spot. So that's a good place to go to be able to determine, what's what.
That's right. So I want to stay on this other diseases too. So there was a question about Cercospora leaf blight, and this person is saying they've seen more Cercospora leaf blight than frog eye leaf spot in recent years. So how do those two diseases interact with each other?
Well, so they are different pathogens. They're both Cercospora, but they are different pathogens that are causing those diseases. And just from talking with, I think Trey Price gave a presentation, gave one of these not that long ago, Dr. Trey Price at Louisiana State University. In Louisiana, that tends to be more of a problem than it is, in Illinois and Kentucky. So, but I do find it, But I usually find that disease a little bit later. So I don't know where that person's from that's asking that question, but there's different disease spectrums that you're going to find that's going to be based on the geography of where you are. And Cercospora leaf blight, it is around, and it can be one of the most prevalent diseases in some places where soybeans are grown.
Right. So I'm going to shift gears a little bit and talk about some of the fungicide resistance aspects and some of the questions that we're getting about those. So one of the questions that we've gotten is, have you tested any of the isolates from these trials that you've gotten to see if there was resistance to the other fungicide classes like the DMIs and the SDHIs?
Yes, we have. In fact, some of that work is funded by the United Soybean Board, and we're doing that work right now. I can tell you from we did some initial work from isolates that were collected from 2008 to 2012, looking at triazoles and thiophanate methyl. So that would be the NBC fungicide, and for the most part, we didn't really see any shifts there. Mm-hmm. We've also done this from 2013 to 2020, and we're still doing this. There are a few isolates that we're finding that are a little bit further on the right spectrum, where they're a little bit less sensitive. And so we need to do some more testing on those to see what that means. If they're less sensitive enough where we would see fungicide failure, I don't know yet, but we're doing that work.
Okay, well, that's good to know, because that proactive approach is going to be really important to farmers as we continue to use fungicides to manage some of these diseases.
And so that's also why it's very important to use, don't just rely on the triazoles, right? Yeah. You have to use two things that are both effective. And so that's going to help slow down the selection as well. Yeah.
Right. And that's a good point, because one of the questions I often get with fungicide resistance, since it always seems to first show up with those QOI fungicides, is that, okay, if we just stop spraying QOI fungicides altogether and just go to these other classes, will we eventually be able to use the QOIs again? Will the pathogen shift and then we can bring those back into the arsenal? Or what are your opinions on that?
That's a good question. Unfortunately, so when something like that happens, that usually means that there's some kind of a fitness cost. So that means that that fungus gave up something else, or is not as good at something else because it's resistant to this particular class of fungicides. And so when you take away that fungicide, then it doesn't survive as well. It doesn't compete with the other ones, right? We're not there's been a lot of work, not just on Cercospora but a lot of work on a lot of different fungi that have developed resistance to this chemistry class. And to my knowledge, there's not really any kind of indication of a fitness cost. So based on that, unfortunately, it's likely that this resistance would still stick around to some degree. Now that, it might normalize, but I think they would still be out there, even if you took away that selection pressure.
Right. So we're going to have to continue to use integrated management, right? Is what that means.
Absolutely.
So there's a good question about that too. So it says, this question is, why are we continuing to use fungicides to control frog-eye leaf spot when there are equally high yielding and resistant varieties available?
That's a great question. So they're not available everywhere, I think. So I think areas where frog-eye leaf spot traditionally has been a problem for a number of years, I think that seed companies have done a really good job of breeding for resistant varieties that have high yield potential. So I think you could do that in some areas. But as you start to see, as we start to see that expansion of frog-eye leaf spot, I doubt that there's very many resistant varieties in a place like North Dakota, or maybe even Northern Iowa. So I think the fungicides are still necessary in places like that. But the other half of that is, this is a pretty variable fungus, and there are different single genes that can be used for host resistance. And already there's been what we call races of the fungus that can overcome that single gene for resistance. So right now there's an RCS3 gene that is used to some degree, that's been highly effective. But if we overuse that, then we could also have a similar problem where we select for races that can overcome that particular gene for resistance. So I think it's important to integrate all these different management practices and not just rely on one single type of practice, because it can, it could lead to failure in the future.
Right. So we have a few more questions. It's right at 2 o'clock, but I'm going to keep going. And so for those of you who might have to jump off, I'm going to ask Kayla to put the link in the chat. If you need CCA credits for this, they're going to be available through the Crop Protection Network. And I see that that link is in there in the chat. So if you need to jump off, you can get your credits by going to that website. But I have just a few more questions, if you can stay on, Carl, because I have a really good one here about fungicide thresholds for application. And so this person says that great presentation. What would be your recommended threshold for fungicide application?
So we kind of addressed that a little bit a few minutes ago, I guess, but we don't have necessarily hard and fast thresholds. It's just hard to do in our crops, I think. And when we think about inoculum levels, and then suddenly a disease can kind of show up. But from my fungicide trials, again, when we see somewhere between 10 and 20% of the leaf area affected, we can have economic yield loss. So I think we want to try to avoid getting there. But a lot of that has to do with, like we mentioned before, susceptible varieties. They're going to differ on how fast they would get there compared to a variety that's going to have a higher level of resistance. So I think it's just a combination of scouting, knowing how susceptible your variety is. If you know you have a really susceptible variety and you're seeing frog-eye leaf spots start to develop at around flowering time, around R1, then that's one that you're probably going to target to maybe spray a fungicide.
Right. I know a lot of people would really like simpler threshold answers, right? But unfortunately, with a lot of our diseases, that's just not an option, is it? So I'm gonna pick your brain a little bit with this question. This person is asking, do you think the bulk of the frog-eye resistance that you're seeing is due to management of the disease, or is it possibly due to the use of fungicides for plant health in the absence of disease?
I don't know the answer to that question. I guess I can answer it by saying anytime that fungal population is exposed to the fungicide, then that selection pressure is occurring. And so that's when we're selecting out those individuals that are less sensitive or resistant to the fungicide. So no matter why they're spraying, if that fungus is present and it's being exposed, that's how this develops. That's the biology. So, I think overuse can contribute to this type of selection. So if we are spraying in times when we don't need to for disease control, then that would increase the selection pressure. So some of that could potentially have something to do with it.
Right. And I thought your slide where you were talking about the soybean foliar pathogens that are not frog-eye, not Cercospora, but are also resistant to some of these fungicides. I mean, that's a little alarming, and I think that should be probably a little bit alarming for some of our farmers. And so what do you think we're going to be doing in the future to be comprehensively managing soybean diseases if we're losing some of these fungicide active ingredients?
Yeah, I think the strobilurin fungicides they're just not going to be very effective anymore for disease control, at least with a lot of these pathogens on soybean. So again, we just have to do those things, kind of like my last slide summarized, I guess, is don't rely just on fungicides. We have a lot of different ways to manage these diseases. So you really have to integrate those different practices so that we're just not putting all the pressure on the fungicides. And when you do decide to make an application, be sure that you have different active ingredients in there from different chemistry classes, and that those classes have efficacy. So if you're using a triazole strobilurin mixture right now, strobilurin's not doing anything. So you're really just using a triazole, right? Mm-hmm. So you need something else in there. So that's something we have to think about. I think especially as a lot of our products go off patent, it may be enticing to choose like a triazole strobilurin mixture that might be off patent. Again, that's only going to be the triazole that's doing the heavy lifting, doing the providing the control. So you need to add something else to it. So I think that's also very important.
Right, especially if you're seeing some small shifts in those populations to triazole sensitivity, right?
Yeah.
I mean, if we're relying on that triazole exclusively and we're already seeing some of those shifts, we could be in a bad situation pretty soon then.
That's a
Yeah.
good point. And with the strobilurins, it was basically overnight. We saw this resistance, right? And that can happen with the strobilurins. It's different with the triazoles, as you know, Kirsten, that that's not going to be an overnight thing. That's a kind of a long-term kind of deal where it's just kind of a slow erosion of efficacy over time. And so that's why it's important that we do monitor over time to see what's happening. And so it's not going to be something we just suddenly see that the triazoles just stopped working. It's going to be that they're just not working as well as they used to. And so that's something we'll have to watch out for.
Right. And so I think that's why the work that you're doing is so important, is because, this is something that maybe isn't front and center in farmers' minds all the time, but it's really important that you're behind the scenes and doing this work over a lot of years. I mean, you have a lot of years of doing this. If you're looking from 2010 till now, I mean, that's a decade of this work, Carl.
I know.
That's a big body of work.
I know. I can't believe we've been doing it this long, but yes, that's true.
Yeah, but it's really important because I think that without that, we're not able to tell farmers, that, these changes are happening and we need to be managing things differently. So I'm going to wrap it up. We have one more question that just came in. And this is, with the expansion of foliar diseases in soybean, are you seeing any potential foliar nematode problems in the Mid-South region?
We have not. We haven't really looked for foliar nematodes. We've looked for nematodes down in the soil affecting roots, but I can't really, I don't have an answer for that question. That's not something I've looked at.
I think it's something that you could be looking at. So you just got a new project today, Carl.
Maybe down the road.
All right, well, that's all we have today. So I just want to thank the Southern IPM Center for hosting it, and Kayla for doing a great job hosting us today. And I also want to thank the United Soybean Board for their support for this webinar series. And also remember to visit the Crop Protection Network if you need credits or just a lot of the information that Carl talked about today. So thank you very much. And I'm going to throw it back to you, Kayla.
And thanks so much, Kirsten, for putting this series together. It's been great. I think people have gotten a lot of great information from it. So thanks for that. Hopefully there'll be another series coming up at some point, but this was the final presentation in the Spring Crop Protection Network webinar series. And I also want to say thanks to Dr. Carl Bradley and Dr. Kirsten Wise at University of Kentucky. Everyone take care, and thanks for attending today.